vaccines have a variety of guises. So some of the early vaccines were known as attenuated vaccines. That is, the infectious organism had been changed such it could still replicate in the human body but not cause disease. And this is what happened with the first vaccine, identified by Jenner, using cowpox as a means of preventing smallpox. And cowpox had sufficient similarity to smallpox. It could elicit, induce an immune response which was protective against smallpox. So that was a whole live virus which was used to prevent smallpox. Later on, people were using what's known as killed vaccines. So influenza is a good example, where influenza is grown up in eggs or tissue culture. It's then killed and then administered as a killed virus to the individual via hypodermic needle. And those proteins are seen as being foreign by the immune system. They're an infectious disease, they don't belong to the body, and therefore an immune response is mounted. More recently, people have been able to identify specific proteins from an infectious organism. A nice example is the hepatitis B vaccine, where the surface protein isolated from the virus via genetic engineering, and just that single protein is grown up, purified, and then used as a vaccine. And just the immune response to that single protein is able to prevent the individual from becoming infected by the virus. So there's numbers of ways that one can generate a vaccine. Tremendous amount of change over the last 20 years. Part of this has been enabled by the molecular biology revolution coming out of the 70s and 80s, which allowed us to clone and express proteins from various infectious diseases, particularly viruses, bacteria, fungi. And being able to work with those proteins has enabled us to get a better understanding as to how those infectious organisms infect individuals, how they evade the immune system. And specifically, it's allowed us to find Achilles heels for the various bugs, whether it will be for antibiotics, targeting various proteins in bacteria to prevent them from replicating, or antiviral drugs, where we can inhibit a viral protein to prevent the virus from replicating in a human cell. The genetics have been very important for us to understand how to make better vaccines against some of these infectious organisms. And really just studying these bugs at a very detailed level and their interaction with the immune system helps us understand how we could perhaps help the immune system do a better job at preventing infection. So generating vaccines that don't utilize the whole infectious organism, just using parts of it, and by generating what we would call a neutralizing immune response, whether it be antibodies which bind specifically to proteins on the bug and effectively take it out of circulation and prevent the person from being infected. The science around infectious diseases has been really quite key to our understanding and give us some really key insights into the immune system. And based on our increased understanding of the immune system over the last 20 odd years has enabled us to discern how we could perhaps use the immune system to manage disease in a way that people probably hadn't thought about only a few years ago. Being so interested in vaccines and seeing how much potential they hold for us, I think we have to see Jenner, who effectively invented vaccination, as being a tremendous stalwart and inventor in the area. So he probably is my number one public health hero. But then we have to go to people like Salk and Sabin, who invented the polio vaccines, and you can go on. But I think it's probably because the vaccines have had such a major impact in improving public health throughout the whole world that one has to look to the vaccinologists as being public health heroes. HIV has been with us a little over 20 years now, and enormous amounts of efforts have gone into trying to find a vaccine against HIV. The situation and our understanding is pretty straightforward because the virus replicates very quickly, but also changes its genetic material, the genes that encode its proteins, on a daily basis. The immune system has a problem on targeting the virus, such that on a daily basis the proteins encoded by the virus change. The immune system, to generate what we call a neutralizing immune response, takes weeks to generate. And so as far as the virus is concerned, it's a moving target. And the immune system really does need a static target, a protein that doesn't change, such that because HIV mutates its genetic material so frequently, so rapidly, the immune system is always several steps behind the virus. And when the immune system does generate an immune response against a virus protein, it kills that virus, but other viruses are selected and are able to replicate because there's no immune response feasible. So the challenges for HIV vaccines are very real and is very noticeable in the respect that we haven't been able to find one over 20 years. However, as we continue to study the virus and the immune system, we're looking for regions of the virus that change less, simply because the function of that protein cannot be changed, and therefore there are conserved regions within those proteins. Now, the virus is clever in the respect that those conserved regions are often hidden from the immune system, but many investigators throughout the world are trying to exploit as best we can what some of those weak points in the viral replication cycle might be. So targeting conserved regions of these HIV proteins may be a way forward. I think our ability to generate better immune responses and faster, so that is antibodies which can neutralize the virus, what we call T cells, white blood cells, able to kill infected cells, getting high enough levels of those types of cells to afford the killing of the infected cells, all very important. And even now people are looking at maybe not targeting necessarily HIV proteins, but proteins provided by the cells that the virus needs to replicate. If some of those proteins could be targeted safely by the immune system, we may prevent the virus from continuing its replication. So whereas perhaps a few years ago people were somewhat daunted by the continuing lack of success in finding an HIV vaccine, typical human perseverance is probably still there, and there are many people trying to find a vaccine. And I'm hopeful that in at least my lifetime we will see a vaccine. It's not beyond the realms of possibility. It just means we need to continue to better understand where the Achilles heels are in this virus. The MRSA bug or bacteria, the methicillin-resistant Staphylococcus aureus organism, is indeed changing, but it's changing from the perspective of its ability to evade antibiotics. So it takes on multiple antibiotic resistance. However, the antibiotic resistance genes that enable this bacteria to avoid antibiotics, and it's a very serious problem because we are running out of antibiotics with which to treat this disease, hence the reason why we'd like to try and generate a vaccine to prevent people from becoming infected with MRSA. Now, those genes that correlate with antibiotic resistance seem to have very little effect on the surface proteins of the bacterium, which are recognized by the immune system. So the challenge for us is to identify those proteins on the surface of the bacteria, which, when the antibodies generated by the immune system bind to the bacterium, can facilitate the removal and the killing of that bacteria from the human being. So the challenge right now, as I say, is to identify those proteins with which we can generate a vaccine to generate a protective immune response. such that people don't become infected with the MRSA, such that they don't need to be treated with antibiotics. Now this is the next great frontier. We've done pretty well over the last century or so to generate prophylactic vaccines, that is, vaccines that prevent infectious diseases. Smallpox has been pretty much eradicated throughout the world. Polio is almost non-existent. Mumps, rubella, etc. There really are diseases of the past. But what we've now learnt about how the immune system works, are we in a position to ask the immune system to recognize what we would call a self-protein, that is, one that doesn't belong to an infectious organism? Our body generates proteins all the time, but our immune system has evolved as we grow up to recognize those proteins as self and doesn't mount an immune response against them. However, one can envisage being able to trick the immune system to recognize some of these proteins as being foreign. So proteins that we associate with disease, we may want to remove from the body via the immune system. So some examples would be, as you mentioned, Alzheimer's disease. We know that in Alzheimer's disease there are proteins which are made or cause the disease, which we would call aberrant proteins. They've been somehow changed from the normal type of protein. And because they've been changed, the immune system can now see that they are different from normal, physically bind to those proteins and remove them from circulation. And indeed there have been clinical studies targeting Alzheimer's causing proteins, and indeed there has been clinical benefit derived from the reduction of these aberrant proteins as a result of the vaccine. So whereas it might sound a little bit far-fetched, because most people associate vaccines with pediatric or infant diseases and preventing infectious diseases, we can envisage how we could harness the immune system to manage certain diseases which are causing real problems in the developed world. Another area that we're looking at doing what we call therapeutic vaccines, that is to treat the disease as opposed to prevent the disease, and that would be in cancer. Now, cancer has some similarities to infectious disease because cancers arise from the genetic changes that are ongoing in our bodies all the time, which generate what we call tumour antigens that are associated with tumour cells. And cancers are effectively arising from cells where their normal state is no longer maintained and they grow uncontrollably. And associated with these uncontrolled cells are associated with known tumour antigens. And by targeting these tumour antigens for the immune system via a vaccine, we can see that the immune system can kill these tumour cells and treat the cancer. So whereas we're early days in the realms of cancer, we do see encouraging progress being made in the clinic. We've had 20-odd years of abject failure in cancer vaccines, but once again, as we understand how tumours are able to evade the immune system and how we can now more specifically upregulate the immune system, we believe that we can help the immune system also cure us from cancer. Vaccines have been with us for, hundreds of years when we look back to smallpox vaccines. And they really have been the spine, the main route for public health management. Vaccines prevent infectious diseases and do so very efficiently. However, there's still some infectious diseases which have been resistant to us to be able to generate a vaccine. And some of the ones that you're all familiar with, like HIV, tuberculosis, malaria, each of those organisms have managed to generate mechanisms with which to evade the human immune system. And yet these diseases have a tremendous toll on human life and morbidity. So whereas we have been successful for many infectious diseases, at least those 3 diseases still require very innovative and different ways of thinking about generating a vaccine. And for each of those areas, encouraging progress is being made both at the research level and also at the clinical development level. Whether those clinical development programs are being run by biopharmaceutical companies or by non-government organisations, encouraging progress is being made certainly against malaria and certainly against TB. Now, if we move beyond infectious diseases and preventative vaccines for those and start to think about therapeutic vaccines and vaccines to treat disease, then that opens up a whole new vista. And what we have learnt about the immune system certainly gives us encouragement that we can generate very specific and hopefully, expectfully, safe ways of being able to harness the immune system to take out aberrant proteins that are associated with disease. And then again, there's encouraging progress on this front. Only last year, a vaccine to manage hypertension showed very encouraging results in the clinic, where a vaccine generated against a protein called angiotensin II was successfully reduced in its concentrations in the circulatory system, and as a result, blood pressure reduction was achieved. So very encouraging progress that we can target human proteins, self-proteins, reduce their concentration and generate the desired clinical benefit. So I think there's great scope for therapeutic vaccines as an alternative modality for managing disease in the future. That's a really difficult question because there are so many diseases which have such high mortality, such high morbidity. HIV is still a tragic disease. Malaria has some of the highest mortality rates for any disease in the world. Tuberculosis. But then you look at Alzheimer's and the morbidity associated with that. Rheumatoid arthritis. All those diseases require cost-effective, easily accessible means of better disease management. And certainly one can envisage vaccines playing an important role in managing those diseases in a different way than we can currently. So if I were to pick one, I probably would have to go with malaria, simply because the death toll from that on a weekly basis far outweighs any of the other any other diseases. Perhaps it is antiquated, but it does work. The virus actually will only grow well in eggs as opposed to other mediums. People are working on growing the virus in tissue culture, that is cells that can also replicate the virus. The various issues with being able to generate enough virus from tissue culture, although great strides are being made in that way. So really what we're doing right now with the flu vaccines is balancing the ability to grow these viruses in the appropriate way. And it just so happens that the virus grows in eggs perhaps better than a lot of other means. However, people are actively looking for ways to improve the amount of vaccine that can be made, and eggs in that respect is certainly limiting. We don't get very large yields from eggs, and as a result people have moved to tissue culture to try to improve yields. And of course there is also now the DNA vaccine technology. And this is using the genes of the influenza virus and administering the gene as DNA into humans, where it is transcribed into proteins and elicits an immune response to the protein from the DNA vaccine. People recognize the shortfalls of having to grow virus in egg, but right now it is still the most effective way of doing it. And as I say, new technologies are being developed to see whether we could improve the ability to generate influenza vaccines more readily. Every year, despite the fact that flu's been around with us since the beginning of time, we learn more and more about flu. We're able to dissect it more effectively, understand its various components as to how it generates disease. Its ability to move from humans to birds to pigs enables it to do what it does, and we are better understanding that process all the time. In our ability to manage disease, though, we have a better idea what we call virulence factors are associated with the virus. There are certain proteins which we know, if are in a strain that is circulating at any given time, are more likely to cause more severe disease. Thankfully, the swine flu currently circulating doesn't have one of those virulent factor proteins. But I think as we understand this virus more and we can better target what I would call generic functions of certain proteins the virus must maintain, it can't change, if we can target those either through antiviral drugs or vaccines such we can generate a universal vaccine, perhaps we will be able to manage the disease more effectively, such that we don't have to generate a new vaccine each year as a new strain of virus comes along. So by targeting conserved proteins or parts of proteins that the virus cannot change just by dint of its function, we may have an opportunity to generate a universal vaccine and therefore not have to worry about the next epidemic, simply because we will have a vaccine that will work against the majority, if not all, the viruses. So genetics is a broad descriptor, but specifically I think we can use the molecular biology revolution, that is the manipulation of genes, and they have been crucial in helping us understand both disease and design better vaccines. So by isolating genes that encode proteins from infectious organisms, we can then take those genes and express them to make new proteins in various different organisms. And that way we can generate very large amounts of protein with which to use as a vaccine. Genetics have also been used as a way of finding new proteins for infectious organisms. Recently, a meningococcal B vaccine has been developed where the whole genome for that bacterium was deciphered and new proteins identified which could be associated or correlated with protective immune responses. And from that identification, again, those proteins were cloned through molecular biology and used as the basis of a vaccine, which is proving very promising, again, in clinical trials. So genetics have been really quite revolutionary with our ability to design vaccines in what I'd like to think both a safe and a very cost-effective manner.